Hello, my fellow viewers. Welcome to Mr.Wanyoike, the digital learning platform. In front of you is Mr. Wanyoike himself, a biology teacher. In today's lesson, we are going to look on gene mutation. Remember previously, we covered on chromosomal mutation. And as far as gene mutation is concerned, this refers to the change that occur in the structure of the gene. And therefore, if the structure of the gene normally experiences any change, it will lead to what we refer to as gene mutation. And as far as the gene is concerned, I said genes are hereditary factors or hereditary units. And therefore, the genes are basically made up of the DNA. And therefore, whenever the structure of the DNA is altered, it leads to alteration in the sequencing of the nitrogenous base. And therefore, once the nitrogenous base sequence is altered, it means that it will lead to the formation of a different codon. And therefore, once a different codon results, it will lead to formation of an intended protein. And once the formation of an intended protein is manufactured, it will translate into different characteristics which were not there in the parent organism. And therefore, gene mutation is very important for us to understand, so that we can know why do things appear different, or the offspring appear different from their parents. And therefore, there are several types of gene mutation. We have the first one. We can look on types of gene mutation. And the first one, we have deletion. That is the first one. And we have the second one is insertion. And the third one is referred to as substitution. And the fourth one is called inversion. And as far as gene mutation is concerned, I would like you to remember the concept of the nitrogenous base. And therefore, I'm going to write the types of nitrogenous base. Base. And previously, I discussed about nitrogenous base, and I said as far as the DNA strand is concerned, we have four basically types of nitrogenous base. We have adenine, we have guanine, we have cytosine, and also diamine. And therefore, the four of them, we have the first one, adenine, denoted by capital A. We have number 2, that is guanine. That is denoted by capital G. We have number 3 is cytosine, denoted by C, and also we have number 4 being diamine, and that is denoted by capital T. And as far as this nitrogenous base sequence is concerned, I'm going to be explaining the types of gene mutation using this nitrogenous base. And from the first one, I can go to the first one. That is, the first one is deletion. And as far as deletion is concerned, deletion is the removal. It involves with the removal of a gene portion, and therefore in a given original DNA strand, therefore we can have the original DNA strand here, having the nitrogenous base. That is assuming that we have adenine, we have diamine, and we have cytosine, and we have adenine once again. And therefore, you can see that here I have original DNA strand. And from this, you will find that one of the gene may be removed. One of the gene is removed. For example, if we remove the original codon, is this one. That is the codon. that is the nitrogenous base which appear in triplet, meaning that they work together all for the they are coded for the information of a particular amino acid, and therefore this is another codon. Those are all original codon. This is another codon. So the original codon, also this is an original codon, and therefore if this becomes the one of the adenine is removed, is deleted, what will happen is that we'll be having the final DNA, the new DNA a strand, and therefore in the new DNA strand we'll be having adenine, we'll be having another adenine, followed by diamine, followed by cytosine, followed by adenine, and therefore you can see that the one of the this one, I can show it here. You can see that this portion has been removed. And therefore, as a result of the removal of the portion, it has changed the nitrogenous codon, nitrogenous base, consequently leading to the change of the codon. You can see that the codon is given by AT, and initially the codon was AAA, and therefore in genetics we normally know AAA is normally coded for the amino acid phenylalanine, and therefore this one is a different codon which is coded for the synthesis of for different amino acid, and therefore it means that due to it has an information for a different amino acid, meaning that a different protein will be synthesized, and as a result of this it will the individual will have suffered from mutation, and that is deletion. And therefore, as far as the second the one is concerned, we have insertion. And as far as insertion is concerned, this is the addition of an extra nitrogenous base onto the existing DNA strand. And therefore, if there is an additional of an extra nitrogenous base onto already existing DNA strand, this kind of gene mutation is referred to as insertion. And therefore, to explain this, we'll be having the original one. Let us take it A, that is the codon, the first codon. Then the second one, T, C, A. And you find that there is a gene G, that is guanine. And this gene comes and it comes, this gene, it is inserted. It comes and becomes inserted here. How will be this is the original? This is the original DNA strand. What about the new one? The new one, how will it be? We'll be having something like this, and the first codon will be A, then followed by G, which is already inserted now. Then you go to A, then you go to another A, then you go to T, then you go to C, and finally to A. And therefore, this is a new DNA, the final DNA strand. It becomes new. Why? Because there is an insertion. The guanine was inserted in between adenine and adenine, and therefore the original one was AAA, coded for the information of the synthesis of a particular protein. What about AGA? AGA is a different codon, and therefore, as a result of insertion, there will be synthesis of a new protein with a different characteristic, and this is what we resort to mutation. The third one will be having substitution, And as far as substitution is concerned, this is the replacement. This is the replacement of a certain nitrogenous base sequence from the original DNA strand, and therefore it is replaced with a different nitrogenous base. And therefore, for instance, we can have the original. This is original DNA strand, and therefore we have a diamine, cytosine, and adenine. And therefore you can find that the G, the letter G, the guanine, this is nitrogenous base. It replaces this, that is the adenine. It is replaced, and therefore it comes and replaces it. And therefore, as a result of replacement, the new one, the new DNA strand, the resulting DNA strand will be like adenine. Then we have a guanine, then followed by adenine, cytosine, diamine, cytosine, and finally adenine. And therefore you can see that the resulting new codon, the first new codon will be AGA. What about the initial one? The initial one was AAA, and therefore AAA is a codon which is coded for the synthesis of a particular protein. What about AGA? It is a different codon, and therefore it is coded for the information of synthesis of a different protein. And this, due to the formation of a different protein, now it will result to what we refer to as mutation. And therefore you can see that after substitution, the original codon was changed from AAA to AGA, and therefore they result to the formation of a different protein which will bring mutation, or that result mutation. The fourth one, and of course the last one, we have inversion. As far as inversion is concerned, a portion of DNA strand carrying the nitrogenous base will cut at 2 points, and the middle point, especially where the DNA strand, where it breaks, the middle piece will rotate through an angle of 180 degrees and rejoin in inverted position, and therefore in so doing it will reverse the nitrogenous base. And therefore an example, we'll be having it, the original one here, that is A. We have an A, we have an A, we have a T, we have a C, and finally we have a G. And therefore, the middle piece, this is the middle piece, you can look at it. This is the middle piece, and therefore the DNA, the original, this is the original DNA strand, will break at point X and Y. Will break at point X and Y, and therefore once it breaks, it rotates through an angle of 180 degrees, and therefore on rejoining, it will form, it will reverse the nitrogenous base. That is, it will become A. Then we have another A, followed by T, because the T was in this side, but after rotation it will reverse the nitrogenous base. We start with T, followed by A, and finally we have C and A following them. So we have it like that, and therefore that is the way that you are supposed to represent it. And therefore you can see that the original codon was AAA for the first codon, and the second one was TCA. That is the second codon. What about in the resulting new DNA strand? How will be the codon? It is AAT, which is different from the first one, and the second codon is ACA, and the original was TCA. And therefore, as a result of the formation of the new codon, as a result of reversing the nitrogenous base sequence, it results to the synthesis of a different protein. it results to the formation of a different protein. It means that the information which is in the DNA will be interpreted by the it will be taken by the RNA, that is the messenger ribonucleic acid. And once it is taken to the where the ribosome is, where the synthesis of amino acid takes place, it means that different amino acid will be combined contrary to the original amino acid, and therefore it results to the formation of a different protein, which will translate into different characteristics, and these different characteristics are what we refer to as to have been brought. We normally say they have been brought by the change in the genetic makeup, and therefore as a result of this, that is the inversion, it will lead to what we refer to as mutation. Having said that, I have come to the end of today's lesson. For more information, you can reach me on YouTube. You search me by the name MR.Wanyoike, and also you can use my number. My number is you can use positive 254. That is the Kenyan code, followed by my number 724 901 639, and therefore you can always reach me using this number for more information, so that I can be in a position to serve you. Till next lesson, bye bye.